And we will pull this over here and there. All right. And we will get okay. Okay. Well, this is my first time live chatting. Oh, there's my camera. Got to look into that. Look into that lily pad. There you go, right there. Now, do we have any questions out there, either through email or online? I'm not sure how to. Let's see here. Top chat. All messages visible. Hi, I'm live for the first time. Here we go. Looks like we might have 10 people on here. So we got a test coming up on Wednesday. So any questions? Anything you have for me? Looks like I've got 13 people, lily pad. Oh, that's your drink. You drink that down. I'll let them wonder what I'm drinking. Okay. Okay. We got several questions here. I'm going to take them first. So Ellie Hudson says, can you explain how cellular membrane has negative and positive charge that creates energy? Does it have to do with the electrons? That's actually a really good question. Okay. So imagine if you have pen and paper in front of you. I don't have a way to draw this, unfortunately. So what you need to do is draw a membrane. Okay, so they have positive and negative charges. It actually has to do with the relative charges. So on a membrane, imagine you're going to pump 3 sodium ions outside the cell. And those sodium ions have 3 positive charges. And you can pump 2 potassiums inside the cell that has 2 positive charges. So what happens is you create a chemical gradient. So I got more sodium on the outside and I got more potassium on the inside. But because I'm pumping out 3 positive charges to the outside, the 2 positive charges on the inside, I build up more positive charges on the outside of the cell, okay? And what that means is because I've got more positive on the outside than on the inside, the positive side is positively charged compared to the inside of the cell. And that's how you get a positive and negative side of the cell. It actually doesn't have to do with the electrons on one side or the other. It has to do with the buildup of cations on one side over the other. Does that help? And because what happens when you build up that electrochemical gradient, that actually stores potential energy. Good? All right, so cool, that makes sense. Now, Marcella, how are proteins formed? Okay, so every single protein in this world is formed inside of a ribosome. Okay, now ribosomes are large molecular complexes made up of proteins and ribosomal RNA. And the protein itself is made up of amino acids. So the ribosome links amino acids together into a protein. And remember, proteins can be formed either in the cytoplasm or they can be formed into the rough ER. And this rough ER is going to make proteins that are going to go out like outside the cell or into another organelle or into a membrane. How does that help, Marcella? Okay, cool. So what's the difference between an anabolic and catabolic reaction? Okay, so our metabolism is the sum of all chemical reactions inside of a cell. Sometimes we build things up. So when we start linking amino acids into proteins, that would be an anabolic reaction. Think like anabolic steroids, those big muscly guys. They're building muscle. Catabolic reactions are when we break things down. So imagine you eat like a steak and you have to take all those proteins and break them down to carbohydrates. You break all the proteins down to amino acids, or you eat like spaghetti, which is full of complex carbohydrates, and you break those down into the individual glucose molecules. That's a catabolic reaction. So anabolic is building. Those are often, don't forget, condensation and dehydration reactions. And catabolic is breaking things down, and that's often hydrolysis reactions. Are we good? Okay, I hope that's good. I'll see if I get another question here. So, Nia, can you explain the fluid mosaic model? Okay. Cellular membranes are made up of mostly phospholipid bilayer, right? But embedded in that phospholipid bilayer are lots of proteins. And in fact, if you were to take a membrane and separate out the phospholipids from the proteins, it's going to be like 50-50 cellular membrane of phospholipids and proteins. Now, the fluid mosaic model is those phospholipids are floating around and they're moving around really, 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 really quickly. And proteins are embedded in them. So it looks like a mosaic of proteins on that membrane. And it's fluid. So imagine a membrane is like a soap bubble, and those proteins are kind of floating around in that soap bubble, hence the name fluid mosaic model. Are we good? Okay, not getting more questions on that one. So Kayla, how are all eukaryotic cells very similar at the molecular level? Excellent question. You know, we're taught from day one that plant and animal cells are really different from each other. But the reality is, you know, these cells all have an endomembrane system. They all have a cell nucleus surrounded by a nuclear envelope. They all have ribosomes that make proteins. They all have an endoplasmic reticulum, both smooth and rough, and they make proteins and rough endoplasmic reticulum. They have Golgi bodies that process proteins. So because of these things, they all share those things in common. That's why they're similar at the molecular level. Okay, any other questions? I think I've gone through the questions I've gotten so far. Turn the music down a little bit there. Can I go over the difference between active and facilitated diffusion? That's another great question. Okay, there are three ways that materials can enter or exit a cell. They can basically diffuse directly across the membranes, so things like oxygen and carbon dioxide, nitrogen, and water. They can diffuse right through the membranes. Okay, but things like ions or electrolytes, they cannot diffuse across the membrane because they can't pass the semi-permeable hydrophobic layer. So here's what happens. We have facilitated transport. And what facilitated transport does, or facilitated diffusion, is we have like an ion channel or a carrier protein. So ion channels allow like electrolytes like sodium, potassium, chloride, or calcium to flow down their concentration gradients, but they have to do it through an ion channel. Okay, now a carrier protein is a protein that will carry glucose across the membrane. They actually grab a hold of it on the outside or the inside of the cell and literally carry it across the membrane. Now, facilitated diffusion does not require an input of energy from the cell. The cell's not spending any work on this. Now, active transport, think sodium potassium pump, calcium pump, proton pumps. We're going to pump a solute against a concentration gradient. So this requires an input of energy, and it's also going to require some type of protein pump to actually do this. And you can think of active transport as like kicking the ball up the hill. You can't just, ball will never just randomly go up a hill. It actually needs something to get it to go up the hill. You need energy in some way to make it go up. So active transport, you got to have the energy and you got to have some protein to help move this thing against its concentration gradient. Okay. I hope that works. Jordan, can you explain how eukaryotic cells came about again? I think the word is endosymbiosis. You got the word right, and that is absolutely correct. Endo means within. Symbiosis means living together. So here's how it happened. About 2 billion years ago, oxygen levels in the atmosphere reached a critical level, like maybe 2 or 3%. That was enough. And life responded to this oxygen by evolving aerobic respiration. That's the ability to use oxygen to break down organic molecules and make lots and lots of ATP from this. Okay. So there were 2 prokaryotes. No proto-eukaryotics, just prokaryotic cells. And aerobically respiring bacteria merged into archaean, the other type of prokaryotic cell. And when it merged, these 2 cells began living together. And originally, the aerobically respiring bacteria is probably a parasite. But over time, that aerobically respiring bacteria evolved a mutualistic relationship. Imagine you and your roommate are best friends versus they're drinking all your milk all the time. In this case, they became best friends, so it became a mutually beneficial situation. Now this aerobically respiring bacteria could produce lots of ATP, and is just allowing the cell to get larger because they have more energy. And eventually, the cells got more and more and more of these aerobically respiring bacteria living inside of them. Those aerobically respiring bacteria evolved into mitochondria, and the surrounding cell also evolved to become larger and more complex with a cytoskeleton and organelles. And eventually, over a certain amount of time, I don't know, tens, hundreds of millions of years, eukaryotic cells evolved from that. Hope that helps out. This is getting pretty full here. Okay, Ariana, can you explain how ion channels work? Okay, that's actually a really good question. So think about this. You have a membrane, and that membrane has a hydrophobic interior. It's got those fatty acid tails. So an electrolyte, which is an ion, it's totally water loving. It can't go through that hydrophobic interior. They don't like each other. So what you do is imagine sticking a pipe through that membrane. And so these ion channels have a hydrophilic interior that allows certain ions to pass through them. All right. And don't forget that those ion channels, they can be gated. You can open and close those ion channels either by changing the voltage or by receiving a signal. Okay. Dan, what is the function of microvilli in the small intestine? Oh, I think I forgot to mention that in class. So microvilli are tiny little projections that come out of the cell. In fact, there's a really good write-up in Wikipedia, or just Google it and you'll see these little things going like this on the bottom side of a cell. Those are microvilli. And what microvilli do is they increase the surface area of the cell for things like excretion and absorption. So inside of your intestines, the microvilli are really, really, really common. And they're there to help your intestines absorb nutrients and energy. So that's why you have microvilli, because you can increase surface area without adding a lot of volume to your cell. Okay, Kayla, can you explain why chemical energy in glucose cannot be converted solely to ATP? Okay, your question is close. Can you explain why chemical energy in glucose cannot be totally converted to ATP? That was a question. Like, let's say, I've got 600 calories worth of energy in glucose. How come I can't make 600 calories of ATP? And that's actually a good question. Remember, there are 2 laws of thermodynamics. The first one, energy can't be created or destroyed. But every time I transfer or transform energy, I lose some of that energy available to do work as heat. So what happens because of that second law of thermodynamics, Hey guys, what happens to that second law of thermodynamics is that I'm transforming energy, so I'm losing some of it to entropy. We're generating heat, which means I can't be 100% efficient at transferring energy. Okay. Hart, what is a polypeptide, and what is the primary structure of a polypeptide determined by? This was a question on one of your quizzes that stumped me. Okay, a polypeptide is basically a sequence of amino acids. You take two or more amino acids and you've got a polypeptide. That means all proteins are by default a polypeptide. But proteins have biological function. So not all polypeptides are proteins, because polypeptides is nothing more than a sequence of amino acids. Let's see here. The primary structure of a polypeptide is, of course, just the sequence of amino acids. Okay. Marissa Moore, can you explain hydrolysis and why it's important again? That's a great question too. Hydrolysis. Hydro means water. Lyse means to split. So if we talk about hydrolysis, that means we're splitting water. Now we've got, when it comes to making our macromolecules, carbohydrates, proteins, and nucleic acids, we build them up through condensation reactions where water comes out of that reaction. When we want to break them down, so if we're going to break down complex carbohydrates inside of our cells, if we're going to break down proteins, nucleic acids, and even fats, to break them down, we have to split water in a hydrolysis reaction. So hydrolysis reactions are also really, really important for catabolic reactions, breaking things down inside of our cells. I hope that answers that. Okay. Holly, 73. Why is the mitochondria not connected to the endomembrane system? All right, fantastic question. The endomembrane system is a series of organelles that are involved in manufacture and breakdown of organic material inside of our cells. The mitochondria are not part of the endomembrane system because they originated through endosymbiosis. So they're part of the energy converting organelles. So mitochondria and chloroplasts are not part of the endomembrane system simply because they aren't directly connected to it or indirectly connected to it, and they evolved through endosymbiosis. They were once free-living bacteria. Okay. Kayla, can you explain the importance of shape for the function of proteins? That's a good question. Okay. Shape matters, form and function. The way something is shaped determines its function. My hand, the way it's shaped, I can grasp things like this drink here. I can manipulate. I can type on a keyboard. There's a lot I can do. If my hand was a paddle, the shape, I could use it to swim. If it had feathers, I could use it to fly. Well, shape also matters for proteins as well. So when it comes to proteins, imagine we have an enzyme and we have to break down the sugar sucrose. Sucrose is a disaccharide made up of glucose and fructose. So we need an enzyme that will specifically fit to sucrose and glucose, just like your hand. You need an enzyme or a glove that will specifically fit to your left hand because it's different from your right hand. And if you don't have the right enzyme, the substrate will never fit to it and the protein won't work. Also, the shape of proteins matter for things like, let's see here, what's a good example? Our pumps. A sodium-potassium pump has to be shaped just right to move sodium and attach the sodium and move it out of the cell and bring in potassium. A calcium pump has to be able to match up to calcium ions versus sodium ions because you don't want to pump sodium ions into your sarcoplasmic reticulum. So like I said, the shape of things totally matters for what they're going to do. Okay. All right, so Jacob, looks like I figured you're out. Marisu, can you explain the endomembrane systems and its importance to the cell? Okay, so the endomembrane system, that's what most of the organelles belong to the endomembrane system. So the smooth and rough endoplasmic reticulum, the sarcoplasmic reticulum, the Golgi bodies, the lysosomes, and all the transport vesicles, and the nuclear envelope. Hey, lily pad. Oh, it's okay. It's okay. Have some milk. No? All right. So all of those parts of the cell form the endomembrane system, and it's really important for packaging and making proteins and shipping them to their final destination. The endomembrane system is also very important for recycling material, so the lysosomes can do autophagy, where they break down and recycle the organelles. They can also break down stuff the cell brings into it. And then the smooth endoplasmic reticulum is important for making things like lipids, including estrogen, testosterone, new membranes, and it's also important for detoxifying things. So like, if you're drinking alcohol, you're going to be producing alcohol dehydrogenase inside your smooth ER. So once again, the endomembrane system is really important for the manufacture and processing of proteins and shipping them to the proper location, and also for building lipids and detoxifying stuff and recycling material inside the cell. Okay. Okay, Jordan, can you explain the cytoskeleton? All right, so we have a skeleton in our body that gives us rigidity and form. It's what we attach our muscles to that helps us move around. Inside of cells, they also have a cytoskeleton, and it's made up of proteins. And with these proteins, they give the cell shape, they anchor the organelles in place, they move the organelles around, they allow the cell to move. They're also involved with things like the flagella, the cilia, cytoplasmic streaming. They separate the chromosomes during mitosis. What else do they do? Oh yeah, the actin filaments are used with muscle movement. And of course, the keratin fibers, these are also involved with giving the cell rigidity, like in the nuclear lamina. So there are three parts of the cytoskeleton. There's the smallest one, which is the actin filaments. Those actin filaments are used in cytoplasmic streaming and in muscle movement, and they're also used in cell division to like pinch the cell off and form two different cells. Keratin fibers are kind of permanent structures of the cell, and keratin fibers, like when you see dead skin flaking off of you, you're looking at mostly keratin fibers, but they make the cells strong and rigid. And then there's microtubules, and these are the largest element of the cytoskeleton. And like actin filaments, they're also modular, which means you can build them up and take them back down again. And then the microtubules are used to like arrange the chromosomes during cell division. Their microtubules are also used to push the cell apart during cell division. And microtubules also are formed in part of the flagella and cilia to help them move. And microtubules also help hold the organelles in place, and they allow like the transport vesicles. They're part of how they move between the rough ER and the Golgi bodies. Okay, can I explain the endomembrane system? I just did that. Okay. Heart. Is the sarcoplasmic reticulum a type of smooth ER, and which part of the cytoskeleton is used in mitosis? Okay, good questions. The sarcoplasmic reticulum is considered smooth ER, but I separated out from the other smooth ER because most smooth ER makes lipids, new membranes, and the steroids, or it's used in detoxification of the cell. So, like your liver has a lot of smooth ER, your testes and ovaries have a lot of smooth ER because they're producing hormones. The sarcoplasmic is a type of smooth ER. So your sarcoplasmic reticulum is found in your muscle cells, and is used to house calcium, which is involved in muscle contraction. When it comes to mitosis, the microtubules are used primarily to arrange the chromosomes in the middle of the cell during mitosis and to pull them apart. And the microtubules also literally push the cell apart as well. And then the actin filaments are used to ratchet down the cell membrane and form two different cells. Okay. Kendall, can you explain how adding salt to water lowers water potential? Okay, great question. Think of water potential as how easy it is for water to move around. Okay, so if you have really high water potential, your water is easy to move. There's nothing holding it back. Now, if I go and add some type of solute to my water, like salt or sugar. It will dissolve in there, but those salts will hold on to the water molecules. So if I have a cellular membrane, salt will try to cross the membranes, but it can't. Salt can't cross membranes. Normally water can, but if I have lowered my water potential, the sodium and chloride ions will hold the water back and prevent it from moving across the membrane. Hence, it has lower water potential. And water will always want to flow from high potential to low potential. So if I keep adding salt to water or sugar or anything like that, or any type of solute, I will lower my water potential, and water will try to move into those regions to reach equilibrium so that the concentrations of solutes will be the same as both sides of your membrane. Okay. Christian, is there a sure way to tell if the substance is low or high potential? That still confuses me and it seems backwards. All right, hold on one second. Yeah, I understand that problem. Whenever we learn osmosis without learning water potential, the issue is we say that water moves from low concentration to high concentration, and that's what confuses people. But the reality of the situation is, think of it in terms of water potential. So the water is going to move from high to low potential. So like I said, adding solutes to it keeps water from moving around, so it can no longer cross the membranes very easily because it's being attracted to the salt ions. So water is going to be where it moves really freely, where there's no solutes in it. It's going to move across that membrane freely, trying to reach equilibrium until there's an equal number of solutes on both sides. Okay. Ellie, are the proteins in the cytoskeleton the same proteins in the cellular membrane? And the answer to that is no. But proteins are one of the 4 types of macromolecules. They're all made up of a primary structure of amino acids. But the sequence of those amino acids ultimately determine the tertiary structure of proteins. So there's tens of thousands of proteins used in a single cell. One type of protein is called actin and makes actin filaments. One type of protein is alpha and beta tubulin. That's actually 2 types of proteins. Those come together to make tubulin. Keratin fibers include several different types of proteins. So the proteins in the cell membrane might include things like protein receptors. They might include those protein pumps, like a sodium-potassium pump. They might include ion channels, like a calcium channel or a sodium channel. While they're still made of the same building blocks of amino acids, they are really, really, really different types of molecules. That was a good question. Okay, Jordan, what do vacuoles do? Okay, so vacuoles, they basically, for our class, what we need to know is they're mostly found in plants, and they store water and minerals for the plant. They have a couple different functions as well, but for us, they mostly just store. They're used for storage. Okay, let's see here. All right. Any other questions? Is that it? Have I answered everybody's question? Oh, there's my live cam. Oh. Is that it? Any other questions here? Are we good? Yep, the test is over 4, 5, and 6. I said this today in class, really make sure you know how to draw those macromolecules. I mean, you're not going to have to draw them on the test, but that really helps you understand them. Review hydrogen covalent ionic bonds. Make sure you understand the difference between active and passive transport. Study the parts of the organelles. You'll need to know their functions as well as where they're located inside of a cell. And make sure you study the theory of endosymbiosis, because that's important as well. All right. Well, I'll give you guys a couple more minutes. If not, I'm going to sign off. Thanks for everybody participating. We had about 20 different people on, so good job. And let me know in class if this works or not. I'm happy to do live chats like this. This is pretty easy for me to do. We had a special guest today, Lily, our special guest star. She's learning lots of biology. I think she's a little sleepy. Okay. Are we done? Heart. Will there be diagrams of molecules that we have to guess on the test? Yes. I will have macromolecules on the test. You will be required to identify them, not just by their name, but also tell me something about what they do. So you have to recognize a molecule and say, yeah, that makes a protein. Yeah, that one holds more potential energy than the other one. Yes, this one is used to store information. So absolutely, you will see macromolecules on the test. You just won't have to draw them. All right, sounds good. Well, alrighty then. I'm going to sign off. If you have any more questions, I will be in my office tomorrow between 3:30 and about 4:30, 5 o'clock. So if you have any more questions, I'll be around then too. All right, you guys have a good one.